Assalamualaikum warahmatullahi wabarakatuh and welcome to our PowerPoint presentation on Chapter 8 Biodiversity under the subtopic of Kingdom Monera, Archaebacteria and Eubacteria. Let's proceed to the next slide. In our PowerPoint, we will firstly introduce what is Kingdom Monera. Second, its classification in taxonomy chart. Third is its usage or applications for the benefits of human, and lastly, its relation to Quran and Hadith. So let's explore the world of Kingdom Monera. You might be wondering, what is Kingdom Monera? So monerans are single-celled organisms that have no nucleus or organelles. They are prokaryotic, which means they contain a cell membrane, cell wall, DNA, plasmids, which is circular pieces of DNA, ribosomes, and cytoplasm containing enzymes. So Kingdom Monera is classified into two groups, which is Archaebacteria and Eubacteria. Archaebacteria are more primitive, being the oldest organism on Earth. They have unique protein-like cell wall which lacking peptidoglycan. Next, their RNA and ribosomal proteins are more like eukaryotes than eubacteria. They also mostly anaerobic and are capable of surviving such extreme environmental conditions such as hot springs and very salty and acidic environments. In this subkingdom, organisms can be classified or grouped on the basis of the environment in which they reside. First is methanogenic bacteria, which is bacteria that can be found in the intestinal tract of animals as well as sewage matter. They are capable of producing methane in hypoxic condition. Next is thermoacidophilic bacteria. These are bacteria that can be found living in hot springs. They are also capable of surviving in environmental conditions with very low pH. These Thermoproteus is the example of thermoacidophilic bacteria. And the last one is halophilic bacteria. This includes bacteria that can be found living in extremely salty condition, for example, bacteria found in the Dead Sea. Next is Eubacteria, the modern prokaryotes. Their DNA is in the form single circular molecule. They do not have membrane-bounded organelles, but possess plasmid and ribosomes. They have cell wall made of peptidoglycan, which is carbohydrate protein complex that protects and gives support to the bacteria. Some bacteria have capsule or a slimy layer outside the wall that provides extra protection for the bacteria. Many bacteria are motile due to the presence of one or more flagella. And some bacteria also possess protein rods called pili. They are shorter and thinner than flagella and function in attachment to the other cell or surface, which is involved in the sexual production of bacteria. There are thousands of different groups of eubacteria, but one of the main groups is cyanobacteria. Cyanobacteria are gram-negative bacteria, formerly known as blue-green algae. Most of them are photosynthetic autotrophs and can fix nitrogen in special cells called heterocysts. So in this picture of lake, we can see a green blue algae here. So that is cyanobacteria. They undergo photosynthesis to make their own food. Eubacteria also include most of the familiar bacteria of medical and economic importance, such as E. coli, Salmonella, and Lactobacillus. Next is their characteristics, which we cover on their structure, shape, and location of flagella. This is the structure of Archaebacteria, while this is the structure of Eubacteria. Flagella located on many parts of bacteria. There are four main locations, which is monotrichous, the presence of single flagella in one end of cell. The second one is bilophotrichous, which is the presence of bundle of flagella in one end of cell. Amphitrichous, presence of single or cluster of flagella at both ends of cell, while diperitrichous is the presence of flagella all over the cell surface. The cell wall that provides extra protection for the bacteria. These are called gram-negative bacteria because these bacteria did not absorb and retain crystal violet stain during lab testing. However, bacteria without the capsule retain the stain and are called gram-positive bacteria. At the end of the experiments, we could see that gram-positive bacteria is colored violet while gram-negative bacteria is pink. There are four main shapes of bacteria. Firstly is coccus. Coccus is spherical in shape. They came in single or a pair or chains or colony. For example, Staphylococcus. And next is bacillus. It's a rod-shaped bacteria. For example, is Streptobacilli or E. coli. Next is spirillum. Spirillum is spiral shaped and usually single cell. For example, is spirochaetes. And lastly is Vibrios, which is comma-shaped and usually single cell. They also undergo several living processes such as respiration, getting nutrition, and their movement. In movement, some Monera can move on their own using flagella. Flagella compose of the protein flagellin and can spin like a propeller, pulling the bacteria through the water. But some Monera cannot move on their own, and they have to depend on something to move them. For respirations, there are two ways. They did aerobic and fermentation. Aerobic needs oxygen, for example, phototrophic aerobe, meaning that there must be a constant supply of oxygen to live. For fermentation, there must be no oxygen around, which oxygen acts as a poison. If there is no oxygen, they use fermentation, and if there is oxygen, they use aerobic respiration. Some monerans are autotroph and some are heterotrophs. Autotrophs meaning that they make their own food, while heterotrophs, they cannot produce their own food, therefore they must obtain it from the environment. The example of autotroph is phototrophic autotrophs, which they gain energy from photosynthesis, and chemotrophic autotroph meaning that they obtain energy from inorganic molecules such as hydrogen sulfide. For lithotrophs are rock eaters which obtain energy by oxidizing inorganic materials, and phototrophic heterotroph are bacteria that take in sunlight for energy, and chemotrophic heterotrophs take in and break down organic molecules. This is the taxonomy chart for Kingdom Monera. The first line is domain and the second one is kingdom, followed by phylum, and expand to its classes until its order. So the blue one here is the order. The first application of bacteria is in the making of antibiotic. For many years, scientists knew that certain molds killed some bacteria. However, researchers needed to understand how to harness this antibacterial microbe and to manufacture enough of the substance before they could make a useful medicine. Penicillin mold naturally produces the antibiotic penicillin, so we extract the penicillin from the mold. Then, scientists learned to grow penicillin mold in deep fermentation tanks by adding a kind of sugar and other ingredients. This process increased the growth of penicillin. So scientists wanted to produce a greater mass of penicillin mold. And then, after obtaining a lot of mold, then scientists separated the penicillin product from the mold. And finally, penicillin is purified for use as an antibiotic medicine. Second application is in the making of yogurt. Firstly, get the milk from cow. Second is sterilize all the equipment to eliminate any unwanted bacteria or any organism. Third is pasteurization. The milk must be heated in 85 until 95 degrees Celsius within 15 to 30 minutes. This is to kill all the microorganisms which originally inhabiting the milk. Fourth is homogenization, which is the breakdown of fat droplets to make them smaller so that they stay suspended in the yogurt instead of sinking and make it all lumpy. Fourth, the milk should be cooled down around 40 to 45 degrees Celsius. After it has cooled down, bacteria is added. Two types of bacteria, which is Streptococcus and Lactobacilli. Milk need to be cooled first to prevent them from being killed by the high temperature. Next is fermentation, which is the changing of lactose to lactic acid by bacteria. Bacteria also start to digest the milk protein. These take many hours and the mixture kept at the optimum temperature of 40 to 45 degrees Celsius so that bacteria can work even faster. pH then dropped to 4.4, that is why natural yogurt has a tangy taste. Next is coagulation, which is the changing form of liquid to solid. After it has changed to liquid, it is time to fill into the containers and ready to package. And the last application of bacteria is in the making of human insulin. So the basic step in recombinant DNA technology is similar for insulin production also. At first, suitable vector plasmid is isolated from E. coli and then it is cut open by restriction endonuclease enzyme. Then the gene of interest is isolated from beta cell and inserted in open plasmid. Plasmid and gene of interest are recombined together by DNA ligase enzyme. And lastly, this recombined plasmid is inserted into suitable host cell, and now this recombined host cell starts producing insulin hormone. This procedure is important to those whose body can produce insulin to regulate the carbohydrate and fat metabolism in their body. This topic is related to Surah Al-An'am, Ayah 38, meaning which the tafsir is, there is not a moving creature on earth nor a bird that flies with its two wings, but are communities like you. We have neglected nothing in the book, then unto their Lord they all shall be gathered. The book in the shade of Quran, Fi Dilal Quran stated, it is an amazing reality which put animals, birds and insects into communities that have their own characteristics, systems and structure as well. The more advanced man's knowledge becomes, this reality becomes increasingly evident. Nevertheless, man's knowledge of a specific creature does not add or change anything to that species. This ayah and these evidently seen facts show Allah's knowledge of the seen and the unseen as well as His planning for everything. This is the list of our reference. We use websites, journals, and books. And that is all for our presentation. Thank you for your attentive listening.